So we ended up right here talking about where does that final electron go to. And in aerobic respiration, it's going to find an oxygen to attach itself to. In anaerobic respiration, it's going to attach itself to pretty much anything that's not carbon-based, but also not oxygen. In fermentation, which we're going to look at now, I just wanted to throw where that final electron acceptor goes, and it does go onto an organic molecule, so a carbon-based molecule. So for fermentation to occur, there does need to be a source of NAD+ that has to go into glycolysis. And so this is the slide we went over when we talked about glycolysis, and we talked about all the things that we got out of glycolysis. And a couple of those things that we get out are NADH, the electron carrier. But to get NADH, we do need some NAD+ to go in. And that fermentation leads us into a little bit of, well, where does this building block go to? Where does this NAD+ that's just ready to pick up electrons come from? And the fermentation process itself provides the source of NAD+. So through fermentation, we're going to gain some of that NAD+ that can then lead to or go into glycolysis and eventually pick up some electrons and become very happy electron carriers. So during the process of fermentation, there is a partial oxidation of sugar. So oxidation, as you recall, is a loss of electrons. So it's really doing a partial breakdown of sugar or some other metabolite to release energy. Because anytime you break a bond, you're going to release energy, and a lot of that is in the form of electrons. And it does use some organic molecule, so some carbon-based molecule inside the cell as its final electron acceptor. A couple of its end products, though, that we get out of fermentation is we do get carbon dioxide, and we also get additional organic products. So we get some other organic products, and part of that is where that electron acceptor is going to end up. Now, the net energy gain is not a lot. Through the fermentation process, we really only get 2 ATP, and that's it. It's not like aerobic respiration that goes through the whole cellular respiration where we can get almost 40 ATP. We're only going to get 2 ATP out of the whole process of fermentation. So as a recall, this big huge box that everything on the right hand or on the left hand side was cellular respiration, this tiny right box on the top was fermentation. And that's what we're going to cover now. And also as a recall, the glycolysis step that hung out right here in the middle, glycolysis is the first step of cellular respiration, but it is also the first step of fermentation. And so the process of fermentation still undergoes glycolysis. So everything that happened in glycolysis that you wrote down on paper, that still is that very first initial step that goes into fermentation as well. Except at the end of glycolysis, you had 2 pyruvic acids, and then they went to make acetyl-CoA into the Krebs cycle into the electron transport chain. Instead, we're going to take these pyruvic acid and we're going to go over into this other side of the table. So here's our pyruvic acid, and depending on what kind of molecule it is, depending on what kind of cell it is, depends a little bit on what they can do with pyruvic acid. There are some cells, some organisms that can take pyruvic acid and make lactic acid. There are some organisms that can take pyruvic acid and ultimately make ethanol. And another slide I have coming up, I'll show some other examples of things that different organisms can take pyruvic acid and make. These are all carbon-based organic molecules as our end products. But if you notice, glycolysis needed some NAD+. Well, once we take that pyruvic acid and break it down and make these other organic molecules, we generate some NAD+. So the NAD+ that's made in this step can go right back into glycolysis and help generate some more of the NADHs that can either go through the cellular respiration if there's oxygen, or can go into the fermentation process in an anaerobic environment. Now, we as humans, what do we do with pyruvic acid in our cells? Well, our cells are going to make lactic acid. And so how you know you've made lactic acid is you're sore after you work out one day. So what ends up happening is that you needed energy, and you needed quick energy for a short amount of time, and your cells were not getting enough oxygen to make lots of ATP as needed. So they were getting some oxygen, but not enough for the amount of ATP that was demanded of the cells at that exact moment. And so what happened is your cells still went through glycolysis, but because they didn't have enough oxygen to go through cellular respiration, they went through the fermentation process. And the pyruvic acid that was made here was turned into lactic acid, and you were sore the next day. However, different organisms can take pyruvic acid and make different products. One example, yeast cells can take pyruvic acid and they can make ethanol. It's our basic alcohol. We cannot do that. Otherwise, if you worked out, you would get drunk, which I guess would make working out more fun. It'd be quite enjoyable and quite entertaining at the gym. But we do not make ethanol. It depends on the different organism, makes different end products with that pyruvic acid. This is showing an example of different organisms and what organic molecules are their end products. So no matter what, it's still going to have glycolysis, and then they're going to get pyruvic acid. Propionibacterium can take pyruvic acid, and through its breakdown, it's going to generate carbon dioxide and something called propionic acid. And this is what's used to give the flavor of Swiss cheese. If you ever wondered why there's holes in Swiss cheese, it's because of the carbon dioxide. Those were actual carbon dioxide air bubbles that formed in there, and the acid gives it its flavor. Aspergillus, Lactobacillus, Streptococcus can all take pyruvic acid, and they can make lactic acid, same thing that our cells can make. And it's usually what gives kind of a sharp flavor to different foods. Saccharomyces can take pyruvic acid, and it can make it'll byproduct carbon dioxide, but it's what makes our ethanol, our wine, and our beer. Clostridium, that same group of bacteria that we can find C. diff at, but different bacteria, can take pyruvic acid and make acetone and isopropyl, the isopropyl rubbing alcohol. So different organisms can take that pyruvic acid and make different end products. They're all organic based end products, but just different end products. It's kind of cool. Now, the last part that I wanted to talk about in this PowerPoint are anabolic pathways. So everything else was really all about catabolic, breaking down carbohydrates, and getting all that energy that we can out of it. Well, an anabolic pathway, this is a synthesis reaction. So we're going to make something and it requires energy. So it's the opposite of catabolic. Catabolic was breaking things down, releasing energy. Anabolic is we're building things up and it requires energy. And where does that energy come from to build things up? Well, it comes from the catabolic reactions. We again are constantly eating foods, our body is breaking them down, it releases the energy, and then we can use that energy to build things up. So it's a constant cycle of breaking things down and building things up. Now, anabolic pathways, the building things ups, are really the reverse of a catabolic pathway. And so a lot of reactions can go both ways. Not all of them. But if a reaction can go either direction, it's known as amphibolic. And so we can take two small molecules, A and B, and we can, through an anabolic reaction, put them together and make a larger structure called AB. But this larger structure could also, in theory, get broken down into two smaller molecules, A and B, by themselves. But there would be a different enzyme for each reaction. So one enzyme would be needed to build something up, one enzyme would be needed to break something down. Not all reactions can do this, but there are some reactions that can go back and forth. Now, kind of one of my last slides here on kind of just some general statements on its integration and regulation of metabolic function. Or really how cells like to be as lazy, efficient as possible. So cells themselves only make enzymes when the substrate is available. So enzymes do something with the substrate. So our cells are not going to make the enzyme if the substrate's not around. That's just too much energy making all these enzymes that aren't getting used. Cells are going to catabolize or break down the more energy efficient choice if two energy sources are available. So if there's two energy efficient choice, two food sources, whether protein, sugar, our cells are always going to go for the one that's going to give us more energy, kind of more of its bang for its buck, the fastest, quickest way to get lots of energy. Cells are going to synthesize or make metabolites that they need when we need them, but then they're going to stop making them if they're around. It's kind of like, if we already have it, why make more? And then we also have what's known as that feedback inhibition that I talked about with competitive and non-competitive inhibition, that ultimately the product, when we start to make a lot of product, that product can be an inhibitor. So that product can go back to an enzyme, it can bind to the enzyme, and then it can stop the cell from making more product. So again, our cells only want to make what we need to make, and then we want to make sure we stop making it once we have it, because that's just a lot of extra energy that's not needed to do all of those processes. And this, I just wanted to show an example. Everything that has a purple arrow is a catabolic pathway. Everything that has a green arrow is an anabolic pathway. No, you don't have to know this, but just to show you that a lot of the products, when you break something down, become a building block that's then used to build something back up. An example, we can take a lipid, we can break it down into glycerol. Well, we just broke something down into a catabolic pathway in the purple arrow, but in an anabolic pathway, we can take glycerol and we can go right back and make a lipid out of it. So a lot of the products can still be used as some type of reactant for building things down. It all comes back to we're constantly breaking things down and building things up depending on what our cells need.